Asked to talk about today is about bioprostheses and biological grafts, and thank you to the previous two speakers about grafts, because again they've done most of the hard work in setting the landscape for me. Some disclosures that I have done some work for various companies, but most of the companies, so it's not a particularly partisan talk. If we look at the evolution of dialysis over the last 60 years has come on from very much a homemade, very clumsy apparatus to something that's very sophisticated, and there's an awful lot of money gone into that because there's an awful lot of money to be made in dialysis. But the evolution of vascular access has been slightly lagging behind, and we've gone from pretty much direct puncture of arteries and veins to a big advance being the Scribner shunt back in the 60s, shortly followed by the AV fistula, which remains the gold standard for vascular access in the current world. But at the same time, there's been attempts to advance the field in terms of biological grafts in the 70s, which I'll cover mainly during this talk, and ePTFE, which you've heard of already in 76. Going back to the previous talk, ePTFE was never really designed to stick needles in, and I always mention to my vascular surgical colleagues that would they be happy for one of my nurses to go and stick a needle in their fem-pop bypass 3 times a week and not expect a problem, and they kind of shy away from that idea. So what are the desirable properties for vascular access? Well, we obviously need to be able to repeatedly needle this and easily needle this. If we can't stick needles in, there's little point in having it. We need to avoid bleeding, it needs to be cheap, it needs to be available, it needs to be durable. We also need infection resistance. As we heard from the last talk, PTFE roughly has a 10 to 15% infection rate, and that's probably why AV access from an autologous option is better. We also need quick maturation. A lot of these patients don't want to be waiting a long time to get their access. As we've heard in the last talk, there are various potentials to try and achieve that with grafts, which ultimately has showed that we can needle slightly quicker but have exactly the same properties of PTFE from the 1970s. I think if asked the question, all of us would turn around and say if we needed vascular access, we would want an AV fistula. However, just see if this works. This paper from Charmaine Locke, which is widely quoted, shows that fistulas are not quite what they're made out to be. Fistulas, certainly, here we are, here's the patency of fistulas over a 3-year period. Fistulas look great, they're much better than grafts, which are terrible. When you actually look at this, this is excluding primary failure. As soon as you include those that fail straight away, fistulas don't look quite so good. And grafts have a much better primary success rate and can be maintained out to around 2 years, and often have better outcomes up to 2 years than fistulas. We may say this is biased data, this is US data, it's not European data. However, these are probably more comparable group with primary grafts. As was mentioned in the earlier talk by Christina, that grafts don't do very well, but we often use them as a second line in patients who are not very well. We wouldn't expect them to do very well. So really, the problems with grafts lie with infection, the number of interventions that we have to do, and ultimately outcomes, but I put that in reference to this graph here. Let's move on to biological grafts, which is what I'm here to talk about. In terms of classification, for those not familiar with the field, it's fairly simple. An autograft is taking tissue from an individual and putting it back into the individual. A good example of this would be taking something like a saphenous vein and using this as an upper arm as a conduit. I'm not going to talk about that because that's not really what we're here for talking about. We're talking about bioprostheses. The next step would be homografts, and this is pretty much the same story as transplantation, taking tissue from one individual and putting it into a genetically different individual. That's what we call a homograft. And then, of course, xenografts are using a non-human species into a human. We'll go through these. Homografts, this is where it all started. In terms of aortic replacements, black and white photographs, because this is historic. But before really Dacron was woven and PTFE was available, people had to find a conduit to replace vessels with. They used homograft from cadaveric tissue. Here's a homograft aorta. This is it shortly afterwards with a big aneurysm having fallen to pieces and degenerated. So it's pioneering, but it's not really durable enough to have any real success. Having said that, there are still situations where we use this. This is a case from my own hospital where we'd removed a large retroperitoneal sarcoma with an IVC resection. Here we used a spiral graft from a cadaveric donor, fresh cadaveric donor, made a spiral graft from the iliocaval segment and used this as an interposition for the IVC. And this patient's still doing very well 3 years afterwards. So it's possible, but not ideal. It's also very reliant on having a good cadaveric donor program to be able to get fresh vessel. This was followed on in the 70s, where rather than take fresh cadaveric tissue, there was an attempt to preserve tissue, and this using freeze-drying tissue, and freeze-drying vein grafts were used. And actually at the time, they were found to be comparable to fresh vein. Equally, cryopreserved femoral veins from cadaveric donors were used and had good primary and secondary patency, comparable to ePTFE. But there's a problem with this, and the problem is that if you are transferring tissue between patients, you will get a sensitization response. What you don't see particularly is rejection, like you see in transplantation, and you get minimal T-cell infiltration and minimal cellular response, but what you do get is an antibody response. And these are renal patients who may well be on the transplant list and may well have problems with later transplantation due to this. And that's a major problem. To try and avoid this, people tried to decellularize the graft. So you take your graft, you treat it in various different ways, take the cells out and create basically a fibrous tube from the graft, which can then be used. The problem with this is the early techniques of decellularization also resulted in degeneration of the tissue, and where actually high rates of aneurysm formation were found again. This was seen in numerous other cases. Some studies showed good outcomes, but other studies showed that high risk of infection, high risk of problems. So therefore people moved away from using decellularized human tissue. But one area that was thought to be of benefit was to use bovine tissue in particular, but xenografts. This is readily available, animals can be treated and can be done on a mass-produced scale, and therefore quality control can be introduced. Still need to decellularize them to remove antigenicity, but they do provide conduits, and we can store these off the shelf. And one of the advantages of these biological grafts is that the local tissues grow into it, and this is 3 months of study looking at bovine tissues, and a vasa vasorum is incorporated into the graft, and actually this helps with infection. One thing that's not noted in these grafts is an endothelial lining, and it still remains a fibrous tube. The different types of bovine tissues used, bovine carotid artery has been used, and some of these results have been very good. 86% secondary patency, 92% secondary patency, and 75% one-year patency, but high complication rates, and there's still dispute. This is a study from 2011 from the Mass General, where actually excellent or good primary patencies were seen using bovine carotid artery as a primary conduit, comparable secondary patencies to PTFE. One of the things to note in this is that PTFE's primary patency at a year was only 10%, so actually is this really comparable? And I think this slide says it all, that it was really no better than PTFE. Bovine mesenteric vein is available under the name of Procol, predominantly in the USA. Primary and secondary patency rates 30% and 58% at 12 months, but these are in difficult patients. The studies are not really there to say this is as good or as bad as PTFE, but similar patencies, so may be useful. Other multi-center trials have shown variable, reasonable patencies comparable to PTFE. Bovine ureter is another conduit that can be used. This is a long, uniform length, uniform diameter tube. Again, decellularized using a variety of techniques. The results of bovine ureter have actually been quite good. And this is a study from Oxford way back in 2011 that showed that this xenograft was actually very good. Eric Chemler did a study comparing with PTFE and again saw comparable results and no difference really between it and PTFE, which as it's more expensive and more difficult to use or more difficult to obtain, really doesn't give it benefit over PTFE. There are also case reports of degeneration. You may have seen this picture in the press. In the literature, big degeneration of a subcutaneous graft, and this is a major problem in the waiting. So there's still question about using bovine tissues, and that led on to the development of using more synthetic tissues, but combining the two. This is the Omniflow 2 that some of you may be familiar with. This is a xenograft from sheep, And what actually happens is a matrix, this polymer matrix, is placed on a mandrel. This is implanted subcutaneously in sheep. Over a time period, this generates a foreign body reaction. Fibroblasts incorporate into forming this tube. That can be then removed, decellularized, and gives you a fibrous tube which is reinforced with a matrix, which can then be implanted. And this is available in different formulations. The benefits of this, actually, there are good results. This is an oldish paper from '96 from Ensler, but it showed that Omniflow, certainly a year, had comparable results to autologous tissue and better than PTFE or bovine tissue, but also with low infection rates. And that's really the key point, is that we can use this in areas where infection might be a risk. Other studies have confirmed this and confirmed similar results with low infection rates and good patencies. And we did a systematic review of this, which again confirmed this with low infection rates. And compared to the PTFE infection rates we've seen at upwards of 15–16%, this is a major advance, because a graft infection is terrible. We don't seem to see the degeneration that you see in other tissues in Omniflow, and it would incorporate into tissues, and it shows good incorporation. Aneurysm formation is low. And probably one of the reasons is this electron microscopy. The matrix, or the scaffold, is here with the fibrous tissue, and this incorporates. And if you look histologically, this is fully incorporated into the native tissues. And we can see here, PTFE grafts may have these areas, and if you're sticking needles through here, get bacteria in there, that's prime site to create a biofilm that you're not going to get rid of. So infection rates are low. If we're looking vascular surgery as a comparison, the Cox study is quite a large one, 2.1% infection rates in peripheral bypass surgery in high-risk patients, and this has been confirmed even in patients using it in an infected field. So what I would say is the advantages of something like an Omniflow biosynthetic graft are, we've got outcomes that are comparable to PTFE, but we have low infection rates, and it may actually be useful in infection patients with Lyme bacteremias, salvaging grafts in infection, and high-risk patients such as skin disease, lower limb grafts, or even for use in TPN. The disadvantages, it's complex to manufacture, which makes it expensive, and it's also the availability historically has been a problem, although that's reduced now. So where do we go with the future, really, to sum things up, is as a surgeon, I'm not really bothered what tube I've got, as long as it is of an adequate size, it's biologically identical to a vessel, it's mechanically identical to a vessel, it's readily available, needs minimal interventions, and it's infection resistant. Now, all of those things are from autologous vein, and something like a basilic vein is an ideal conduit for this. But also in the future, we may be able to use tissue-engineered grafts or tissue-engineered blood vessels. And just to quickly run through that, there are something like about 75 patents already filed for tissue-engineered grafts. But at the moment, the two that are interesting are the humocyte graft, which is made in bioreactors in the States, available off the shelf with long preservation time and can be implanted. Scaffolds used, human cells, human cadaveric cells are implanted in this, and there's a mechanical stimulus in here to make it stronger, and this can be used to produce a biological tube. Again, this is simply a fibrous biological tube. It's not cellular, it's not a blood vessel. And you can see that it's collagen and other tissues. It can be used, and it has been used in animal models. The bigger question would be, can we actually make a vessel that's individual to a patient? And we can. Can we make it biologically and mechanically compatible and off the shelf? So we can use the humocyte and implant patient cells in the bioreactor, which create an endothelial surface. Or more interestingly, although experimentally, you may be aware of Joris Rotmans from the Netherlands, who's doing this study, who you use your biological scaffold, you implant it under the skin, and this is in an animal model. This then has a fibrous reaction. You can remove this. You can then subsequently reimplant this, and this is in a carotid model. After 4 weeks after grafting, what actually happens is this turns from a fibrous tube into an arterial tube, and these are smooth muscle cells that have infiltrated the graft and started to remodel the graft to look more like an artery. The other thing that's quite interesting is, if we look at the lectin, it actually retains an endothelial surface. So this may well remodel in the same way as a vessel would remodel, which in itself may have problems. So just summarizing from that, What are the advantages of biological and biosynthetic grafts? One might be the biological and mechanical properties. One might be the infection properties. But one of the problems we see with a plastic tube is that it creates a lot of hemodynamic problems at the outflow, and this is where we see problems, and it may be that that mismatch in mechanical outflow assists in maturation. So biological grafts have had a long history of complications which have made them not competitive with PTFE. It may be that we are moving into an era where biological grafts are competitive with lower infection rates. This is all very exciting, but ultimately what most of these products are trying to make is creating the histological structure of an artificial artery. Yet the gold standard in vascular access is an autologous vein, and perhaps we're looking at the wrong direction in terms of doing this, because we'll get the problems of sticking needles in arteries and with near-intimal hyperplasia. We still don't have a perfect conduit for dialysis, and that also includes AV fistulas. Thank you very much.